First of all, thank you very much, Carolina, and the other organizers for the invitation. I would have loved to be in Brazil, but this is life. So I will talk about optimizing sensory feedback in a brain-machine interface in rodents. So for those of you that have been yesterday in Dr. Shakur's talk, he was talking about neuroprosthetics and proposed a modular strategy in which neuroprosthetics are defined by three fundamental blocks, which are the motor decoding, the movement restoration, and the sensory feedback restitution. And I will be, so that has been done by Shakur in a sort of approach, a neuroengineering approach. And I will talk more on, I would say, neurobiologist perspective for this sensory feedback, okay? Okay, so let me start by showing one example in the visual system, and then we'll move to the tactile system. What you are probably experiencing here is the scene of a continuous movement as a sort of worm composed of three red dots, okay? So this is a very powerful illusion, in fact. This is what is called the beta phenomenon, or the apparent motion illusion, when there is a visual target that is flashed successively and immobile. This is very important in different positions in the visual field. You get the impression that this is a moving object, okay? Although at each time the image that is presented is stationary. The subject yourself, you can perceive a continuous motion, okay, of the object along a trajectory here along the sides of this square. So this beta phenomenon has been described by Max Wertheimer in the early 90s, 1912 exactly. He was one of the founders of the Gestalt School of Psychology, and he proposed a theory of perceptual grouping where the experienced percept contains more explicit spatial information than the sensory stimulus on which it is based. Now, in the tactile domain, this is, can be also the case, because in tactile perception, like in this picture, touch relies on the ability of the system to encode distributed information, distributed inputs on certain parts of the fingers here, or the palm, but you perceive tactilely a whole object, okay? You can give out of this partial information, you can restore the entire, for example, form, shape, rigidity of this object. Okay, so my talk today will be in two parts. The first concerns the neuronal basis of this kind of perceptual phenomenon. And the second part is whether we can use this information that we gain about sensory processing to provide sensory feedback in the context of a closed loop brain-machine interface. Now, in my lab, we use the whisker-to-barrel cortex system in rodents as a system model, and this system has become a dominant model to investigate sensory-motor integration. And this model allows a multi-scale approach. We can go from cellular to network to behavior levels using different kinds of techniques like electrophysiology, imaging, operant conditioning, etc. So let me give you just a glance of what are we talking about when we studied this system. So this system starts in the whiskers on the moustachial pad on the snout of the animal. There are 30 in rodents. More or less 35 whiskers on each side of the snouts. And this information that is gathered by the whiskers is transmitted to the barrel cortex, where you can see here an image, just a histology of the barrel cortex. And I here delineated the contours of this map. As you can see, I call this the retunculus. And as you can see, the representation of the whiskers is right there. It's very, very enlarged. There is a magnification index here that is very important. It's giving you the idea of how important this system for the survival of the animal. So what is nice in the system is that there is an ordered topographic representation of the whiskers on the snow in the barrel cortex. So this somatotopic organization is preserved from the pad to the cortex and contains a full surface, a full representation of the surface of the animal. Now, the classical description of this system, it's a sort of label line projection. That is, you have a certain number of whiskers on the snow, a certain number of barrels in the cortex, in the somatosensory cortex, and there is a very nice correspondence between each whisker and each barrel in the cortex. So that if you want to know what happens in D2 whisker, for example, all you have to know is what is the activity in the D2 barrel. This is the way this system has been described for the last 50 years. But at odds with this canonical description, there are many anatomical, behavioral, functional evidences that the system integrates multi-whisker information when the animal is engaged in, for example, contacting an object. Okay. And so we were very much interested in this idea that the system is using multi-whisker information. And for that, we built, it took to us 5 years to build a matrix that allows us to stimulate 25 whiskers at the same time on one side of the snout of the animal. This case is a rat, but we have also the same machine for mice. This is based on piezoelectric benders. You can contact the whisker within each one of these small plastic tubes, and you can move the whiskers down to 1 micrometer in a very repetitive way. So using this matrix, we have conducted a comprehensive study on the dependence of cortical coding on the input statistics, because with this machine, we can control very precisely the input to the system, of course, in the anesthetized animal for the moment. So using this, we tested sequences of rostrocaudal whisker deflections that generate multiple bristle motion patterns, as you can see here in this small animation there. So, of course, this is a visual representation of the actual tactile stimulus that is given to the whiskers on this note. And we recorded activity, neuronal activity in the C2 barrel, and this kind of stimulation allows to disambiguate local from global sensory integration, because locally each whisker has been deflected in the same way. All the movements are caudal. But with this, we can generate sequences that in 8 different directions that are depicted here. So you have to imagine it's like you move an object across all the whiskers in different directions, and then you record activity in C2 barrel. If C2 neurons were only caring about the C2 whisker, then the neuron would not be able to extract any information about the global motion. Whereas if it integrates multi-whisker information, then eventually it can give some information about the global motion. And this is indeed the case. This is just one example. You can see here that the response of the neuron is increased for this caudal movement, whereas other movements in other directions give rise to a much smaller activity, so that you can build a tuning curve for apparent global motion in the C2 barrel. And this is the average vector for this particular neuron. Now, this is not just one neuron. It happens in most of the neurons in the cortex. 65% of excitatory neurons, 81% of inhibitory neurons show this selectivity to apparent global motion. And if you take now the preferred direction for all the neurons that have been recorded in the C2 barrel, this is the distribution, and indicating that each neuron has a preference for a given direction. But if you take the overall activity in the C2 barrel, then you can extract a sort of average vector, meaning that if you consider the whole activity in C2, all the neurons together, they are pointing to this downward and leftward direction. This is very reminiscent of the seminal work of Georgopoulos on neuronal population coding in the motor cortex in monkeys, just that this is the sensory cortex. Okay. Now, at the population level, what we observe is that in the C2 barrel, there is sort of a vector pointing down and left, but we also studied the activity in other barrels, like this alpha or beta, and we observed that these barrels are selective for a different direction. And this led us to make the hypothesis that global direction selectivity could be represented or spatially distributed at a suprabarrel scale over the barrel cortex. And for this, what we have done is to combine the matrix that I showed you before with voltage sensitive dye imaging of the somatosensory cortex in mice. So we moved from a microscopic electrophysiological approach to a mesoscopic analysis of the apparent motion. And the stimulation protocol was exactly the same. So this is the overall results indicating that there is indeed a suprabarrel map of direction selectivity to global motion in the mouse somatosensory cortex. I hope this will work. These are small movies indicating giving you the activity overall over the barrel cortex for the different global directions. I hope this is working. This is not. Can you see that? Because I cannot. Carolina? Okay,
Sorry?
in any case.
Yes.
Can you see a movie there or not?
No, no.
Okay, so I'm sorry, it doesn't work. Anyway, it worked a few minutes ago. So what I was just seeing here is the activity for the different movements, and out of this activity on the barrel cortex, we can extract the average vector for each one of the barrels. And as you can see here, there is a global sort of preference for the caudoventral global motions. This is very interesting ethologically because when an animal is moving along a wall, this is the kind of stimulation that will be generated by all the different whiskers, okay? So in any case, you can see a gradient through the barrel field of global direction selectivity. So as a sum up of the first part of my talk, I just show you that barrel cortex neurons respond to a wide repertoire of whisker movement features. I didn't really show you, but they respond to selectivity to inter-whisker correlations such that when the animal contacts an object or detects an object with all the whiskers, there is a strong response in the barrel cortex. And I also show you that these neurons code for high order multi-whisker information, and that there is a suprabarrel map for these global motions. So until now, we showed that the system can extract information on global properties of the stimulus, a sort of gestalt, if you may, but this has been done under anesthesia. Now, the question now is whether if the animal can make use of this information, for example, whether the animal can detect a global coherent stimulation of several barrels and produce an overt behavior accordingly that we can record from an awake animal. And this is what we have done next, in which we first, instead of using the stimulation of the vibrissa using the matrix, which is very, very difficult in the awake animal, we used optogenetic stimulation of the somatosensory cortex in a head-posted awake animal. These are animals, AI27, EMX Cree mice that express channel rhodopsin and excitatory neurons. And the first test that we did is that we checked if the animal can detect the optogenetic stimulation and show a detection through an overt behavior, in this case, leaking for obtaining water reward. And it isn't the case. If you shine light, blue light on the somatosensory cortex, the animal increases the rates of leaking. Whereas if you move the stimulation to away of the opening here to the skull, then there is almost no response as shown here in the sham. Okay, so now we are sure that the animal is able to detect the activation of the somatosensory cortex through optogenetic stimulation. Now, we submitted these animals to a learning which is quite complicated. We shine a barrel of light, blue light, on the barrel cortex. These are circular trajectories, can go in which direction. And we presented more than 250 different trajectories that we recorded from other animals, so that these animals cannot know in advance what is exactly the movement of this light that is shining over the somatosensory cortex. Now, we defined a starting point that is always here down, and then an area which we call a no-leak zone. That meaning that when the stimulus is in this position, in this area here, in the red area, if the animal leaks, then there is a punishment, there is a time out of 5 seconds in which the trial is stopped. Now, if the animal leaks when the light is on these two sides, nothing happens. But if the animal leaks when the stimulus is in the green zone, then he gets, in fact, a water reward. So what is this? Is this animal able to learn such a complex stimulation? Well, the answer is yes, otherwise I wouldn't show you that. I'm sorry for this slide, it's a little bit complicated. These are trials, these are more than 20 trials. Each line is a trial. The red here, or the pink here, it corresponds to the area there. This is against time. And each time the animal is leaking in the red zone, you see there is the trial is aborted, and there is a gray line here of 5 seconds, okay? So at the session 1, when the animal starts learning, almost all of the trials are stopped, and because the animal didn't do the right thing. Whereas 5 days afterwards, as you can see, the animal never leaks when the stimulus is within the non-leak zone. It starts leaking either in the buffer zone or in the reward zone, and the number of rewarded trials is significantly increasing across sessions. And also the detection rate. The detection rate of the value is the percentage of entries into the rewarded zone for which the mouse obtained at least one reward. So overall, a high detection value indicates that the mouse learned to leak when the light bar was in the rewarded zone, while a high percentage here of rewarded trials requires that, in addition, the mouse learned to refrain from leaking when the stimulus was in the no leak zone. The next question we asked was if there is something special about the barrel cortex in this task. So we wanted to know if the spatial organization of the barrel cortex was key in this sensory guided learning. And we tested 2 more areas. One belonged to the primary somatosensory cortex, but it was in the representation of the trunk of the limbs and the snouts, but not of the whiskers. And the second area was here, the posterior parietal cortex, which is a non-tactile area with no strong topography. And as you can see here, for both the somatosensory cortex outside of the barrel cortex, as well as for the posterior parietal cortex, there is no learning. So we, in fact, conclude that the continuity of the somatosensory cortical maps in the barrel field favors the learning of a sensory guided behavior. And this is important because if you want to apply this in a closed loop brain-machine interface, you have to take this into consideration. And this is what we have done next. And this is starting the second part of my talk, in which we have a closed loop approach to probe the brain and to try to understand if we can use the knowledge that we acquired in the previous experiments to inject sensory feedback in this BMI. Now, we know that sensory feedback is necessary because we know that there is a degradation of the motor control in the absence of sensory feedback. For example, if you inject lidocaine in the tips of the fingers, it's very difficult, although motorically there is no problem, but it's very, very difficult to hold objects and to do precise movements. On the other hand, it has already been recognized too that in the field of motor learning, that the feedback can be detrimental for the learning process if it is not provided properly or correctly. So this is why the experiments that we did before are so important to implement a good sensory feedback into the brain-machine interface. So what we did is that we did chronic recordings in the primary motor cortex of mice. These mice were awake and they could get a water reward. With this neuronal activity in the motor cortex, we generated a motor command that can either control, for example, a cursor on a screen towards a given target, or it can actually, so this is virtual, of course, or it can actually control a robotic processes. In this case, it was just a 1D, 1 degree of freedom processes. And the location, in fact, the position or the velocity, in fact, of this target, of this cursor or the processes were fed back into the somatosensory cortex through a display system for optogenetic activation. And so for the recordings of spiking activity in motor cortex, we use silicon tetrodes, and so we have many tens of neurons recorded simultaneously. We have a way of doing spike sorting online, with very small delay of a few milliseconds, and then we can choose out of this many neurons recorded simultaneously. We choose 3, and we use these 3 sort of master neurons to control a bar, in this case a virtual bar that is moving across the whiskers, as I showed you before in the electrophysiological recordings. So here we have a bar that is moving along the pad. It's contacting virtually the whiskers, and it's moving on left to right. And there is one position that, as you will see, is the position where the animal can get the reward. I will come back to this in a minute. I'm sorry, I blocked this for some reason. I'm sorry, Carolina, I don't know if you can hear me. This has been blocked.
Yes, we can hear you, but it's blocked here too.
Yeah, I don't know why. Okay. Oh, here we go. I'm sorry. Okay, here we go. I'm sorry for that. Okay, so on the other hand, we have to build the sensory feedback, right, to stimulate a somatosensory cortex. And so the position, the actual position of the light bar, was fed back into the somatosensory cortex as light. Okay, so we had sort of barrel-aligned photostimulation patterns through a projector, and we could stimulate individually each one of the barrels on the somatosensory cortex. Initially, we do a functional localization of these barrels by using intrinsic signals, and once we localize the barrels, then we can stimulate them with these blue lights, okay, individually. And the way we did this is that we correlated the position of the stimulation in the somatosensory cortex with the activity in M1, so that the activity is very low. The bar light, the virtual bar light, is on the left side, and we stimulate these. This is the actual stimulation. We stimulate the barrels on this side. If the activity in M1 is increasing to the other side, to the right, then we stimulate the right-hand barrels. But if the activity in M1 is maintained within this middle area here, then the bar is entering the rewarding zone, and then when the animal leaks, he can get a water reward. So he can only get reward in this if he controls the motor activity to put the bar in this actual position. And this is precisely what the animal does. As you can see here, this is the number of rewards per second. And if you use this biomimetic feedback, that is this very well-structured lines of stimulation, there is an increase in the performance, both in the rewards, number of rewards per second, or a number of percentage of rewarded leaks. Whereas if you do not, you open the loop and you do not give this sensory feedback to the animal is unable to solve this problem. Now, interestingly, if we shuffle the stimulation, that is, if we maintain the very same number of barrels being stimulated, but this time we shuffle them spatially, and we keep the same relationship with respect to the activity in M1, as you can see, there is no learning in the shuffle condition, whereas there is learning here in some more, these are different animals than the ones I showed you before. In the biomimetic case, there is still an increased activity. Carolina, I don't know how good I'm doing, but I have 1 or 2 more slides and that's it.
You can go on, it's perfect.
Okay, so until now, to sum up, what I show you is that there is selectivity in the barrel cortex to high-order multi-whisker information. These neurons respond to a wide repertoire of whisker movement features, and the animal, as I show you here, can make use of this information to produce an overt behavior, and this sensory information is indeed needed in a closed-loop condition to produce optimal motor commands. I will finish with this slide, which is one of the Zoom that we did lastly with my team. And there are 8 different nationalities here, from Russia, Morocco, USA, India, Argentina, and France, obviously. And I want to thank particularly Valérie Gostengel, Isabelle Ferezou, and Lucas Estebanez, who are 3 tenure researchers in my team and who are responsible for most of the experimental work that I've just showed you a few minutes ago. And let me finish with this. I cannot refrain myself for showing you this very brand new building in which we moved a week ago. So it's a very exciting place to be. It's 30 kilometers southwest from the Eiffel Tower. And of course, there are many possibilities here for postdocs. If you're interested, you have to enter this website of the Institute. And thank you very much. I will take questions now.
Thank you very much, Daniel. Really interesting and impressive results and work. It can be seen that, well, you have an amazing team.
Yeah, definitely.
So we have a question here from Joseph. He said, excellent presentation, Professor Shulz. I have a doubt. What are the challenges of translating the results you achieve in animals to a brain-computer interface or either brain-computer interface control neuroprosthetics with sensory feedback in humans?
Yeah, it's the $1 million question. So there are several answers to that. Of course, for the moment, the feedback that can be given back to humans is using techniques like intracranial microstimulation, which is something that is used in humans already for many, many years. There are other ways of giving a feedback to the humans, but indeed, I would say that optogenetic stimulation is far from being implemented in humans. Although in Paris, in the Institute of the Vision Institute, they are very, very much advanced in using optogenetics for blind people in retina. So these are injections in the eye, and the retina is expressing these channel rhodopsins, and now they can stimulate directly the retina with this technique. So I don't know how much we can extrapolate this and say that in 10 or 20 years from now, we will have the ability to, very locally inject viruses in a somatosensory cortex. I cannot make even a guess about this. But the idea here was to try to describe as much as possible the sensory feedback that is needed to optimize the use of a BMI. We cannot think that a so sophisticated tool like the brain will accept any somatosensory feedback. Okay, so we have to be careful about that. And the only possibility that we have today is to use either rodents or monkeys, of course, and we are exploring the possibility of having here marmosets in the institute to do this kind of somehow scaling up in the evolution line, towards humans.
Here we have, Daniel, a lot of congratulations messages from Sadie, Leah. Thank you very much. Really interesting presentation. And we have more questions. You have time?
Of course.
We have one from Sadie. How do you think the specificity of cortical localization of stimulation would translate to human subjects?
Well, I think that it will fully translate. I think that, as you know, I talked about the retonculus as an homology for the homunculus, right? So there is an homunculus in the brain, and so the areas that receive information from the fingers, for example, are very well localized in the human brain. So we can think of aiming those areas if we have the technique. But even if we use intracranial microstimulation, we can aim those areas of the representation of the fingers, for example. Of course, okay, so this is more a philosophical way of answering that question, is whether we have equipotentiality in the brain so that we can use any area of the somatosensory cortex, for example, to give back feedback. I've been working on plasticity in the cortex for decades, and I know that the cortex is extremely plastic. So I guess that if we insist, probably we can convince the brain to modify itself to receive somatosensory feedback information in different areas. But I think that optimizing the localization of the stimulation can accelerate the acceptance and the use of BMIs, which today it's a real problem. I mean, subjects are not really happy, with this brain-machine interfaces. And one of the main problems is that they don't get the optimal sensory feedback.
And alumno Joseph pregunta, Daniel, Joseph is asking do you think that optogenetics is the future of brain-computer interface?
Hard to say. I think it's extremely powerful. I think that we are using for vehiculating this channel rhodopsin viruses that can be used in humans. So yeah, the main problem might be ethical. I mean, can we use these viruses directly injecting this into the brain of humans? I'm not so sure. Who knows? I mean, maybe we will find a way of stimulating from outside, because one of the main problems of intracranial microstimulation and optogenetic stimulation, I mean, viral injection, is that these are very invasive techniques. So I'm not sure that everyone will accept this kind of strategy, So probably the field will move towards trying to find more non-invasive ways of stimulating, like transcranial magnetic stimulation, that for the moment it's okay, but I think that this technique has to be improved. I mean, the spatial definition of the stimulation is not enough, fine enough, let's say, to control the sensory stimulation like we did here with optogenetics.
Thanks, Daniel.
Sure.
So let me see if we have some more questions here. So we have congratulations messages.
Thank you.
Well, Daniel, it was a really impressive work. Thank you very much for being here with us.